All right, everyone. Settle down, please. Let's get started. Today, we're diving into a really fascinating topic from your textbook, Chapter 8, Part 1. We are going inside the human body, but not as doctors. Oh no. We're going in as a tiny, single-celled invader on a grand mission. We are going to uncover the secret battle plan that every successful pathogen follows to cause disease.
So this is the big question, isn't it? How can something so small, something you literally cannot even see, manage to take down a system as incredibly complex and massive as a human being? I'll tell you, it's not by accident. It is a calculated, evolutionary masterpiece of warfare.
So I want you to think of this less like a dry lecture and more like we're following a military campaign. We're going to track our little microbe from the moment it first breaches our defenses all the way to its final destructive victory. Come on, let's jump right in.
Okay, so first things first. The invader has to get inside, right? Our body, you see, it's like a fortress. It has high walls, guards at every single gate. But these pathogens, they are very, very clever saboteurs. Now look at this table. You must understand that these microbes are specialists. They don't just attack anywhere. They have specific strategies for each entry point. To get through your skin, they might just wait for a small cut or an insect bite to give them an open door. In the lungs, they release toxins to paralyze our tiny sweeping brooms, the cilia. And in the gut, my goodness, in the gut they have to survive a literal bath of acid. So for every defense we have, a clever pathogen has already found a way to get around it. Let's just focus on the gut for a moment. It's an incredibly hostile place. That gastric acid is no joke, Beta. It's a powerful chemical barrier designed to absolutely obliterate almost anything we swallow. It's one of our most important defenses. But what happens if that defense just fails? Look at that number. Just look at it. 10,000 times. This shows you so perfectly just how crucial a single defense is. For the cholera bacterium, just a little bit of neutralization of that stomach acid, maybe from some food or an antacid, and its chances of causing a deadly infection increase by 10,000-fold. It's a game of exploiting the smallest weakness.
Okay, so our invader is in. It has established what we call a beachhead. But one location, that's not enough. The mission is to conquer the whole body. So the next question is, how does it travel? How does it get from that first point of entry to organs far, far away?
Well, the body itself provides the transport network. The bloodstream, that's the superhighway, the fastest and most common way to get anywhere. The lymphatics are like a parallel road system, often used by bacteria like M. tuberculosis. And then you have the really sneaky ones, like the rabies virus. They hijack our own nerves, a stealthy, hidden path that leads straight to the central nervous system. So the plan is quite simple and logical, you see. Step 1, get in. Step 2, multiply your forces, and this happens fast. Some bacteria like E. coli can double their entire population in just 20 minutes. After that, step 3, get on the highway. And finally, step 4, establish new colonies, seeding infection in distant organs like the liver, the heart, or the brain. And you know, this widespread invasion has a terrifying name, sepsis. When these pathogens are just everywhere in the blood, the body's immune system can go completely haywire. This leads to a catastrophic body-wide inflammation that can cause organ failure and, sadly, death. This is the real danger of a successful spread. Now, this is the really clever part. This is where it gets interesting. Once inside, the microbe has to deal with our internal security force, the immune system. And these pathogens, I tell you, they have evolved these incredible espionage tactics to hide and even disable our cellular police force. One of the most brilliant strategies is something called antigenic variation. Just imagine our immune system is looking for a criminal who is wearing a red coat. Well, the moment our body learns to spot that red coat, the pathogen, poof, it simply changes into a blue coat. It is constantly changing its appearance, its surface proteins, so our immune system can never get a proper lock on it. The influenza virus is a master of this trick.
And their toolkit is vast. Some bacteria you see here, they build a slippery capsule to resist being eaten, that process we call phagocytosis. Others can go completely dormant, hiding for years in a state of latency. And some, like Mycobacterium tuberculosis, are just so audacious. They get eaten by our immune cells on purpose. But then, they disable the cell's digestive machinery, turning their would-be prison into a 5-star hotel where they can multiply in safety.
Yes, this concept of latency is absolutely fascinating. Take the chickenpox virus, for example. After you recover as a child, the virus doesn't leave your body. No, it retreats into your nerve cells and basically goes to sleep. It lies low, expressing almost no genes, for decades sometimes. Then when you're older and maybe your immunity is a bit weaker, it wakes up and reemerges as shingles, a true ghost in the machine.
So our invader is in, it has spread all over, and it has evaded the immune system. The final stage is the damage itself, the symptoms, the disease. So the crucial point is, how does this damage actually happen? And let me tell you, it's not always what you think.
So bacteria have two main types of weapons, endotoxins and exotoxins. Endotoxin is quite simple. It's part of the bacterium's body, its cell wall. When the bacterium dies and breaks apart, this endotoxin is released, and it sends our immune system into a complete panic, causing fever and shock. Exotoxins, on the other hand, are like smart bombs. They are proteins that are actively fired out by the bacteria to perform a specific mission, like the botulism toxin that causes paralysis.
So really, we have three main ways the damage is done. First, there's the direct attack, where the microbe itself is the killer. Second, the toxin release we just talked about. But the third one, friendly fire, is perhaps the most important concept to grasp. Often, the worst damage is not caused by the microbe at all, but by our own immune system's massive and sometimes clumsy response as it tries to destroy the invader.
And that leaves us with a final, really thought-provoking question. When you have a fever, a cough, inflammation, when you feel sick, is it the germ that's making you feel that way? Or is it the collateral damage from the furious battle being waged by your own body's defenders? Something to think about, isn't it? All right, please review this chapter before our next class. That's all for today.
Hello, everyone, and welcome back. So today, we're going to dive into the very fascinating and, let's be honest, sometimes quite scary world of viruses. We're going to look into their battle plans, okay? How they infect us, what clever tricks they use to hide from our immune system, and most importantly for us, what are the microscopic clues they leave behind for pathologists like us to find?
Right, so here is our agenda for today. A quick roadmap. We'll start with the basics, the rules of spread, then we'll look at a pandemic story we all know, COVID-19. After that, some childhood classics, measles and mumps. Then these clever sleeper agent viruses, and finally, we'll wrap it all up with the key lessons. Simple.
Okay, so first up, sexually transmitted infections. Now look, we are not just discussing STIs for the sake of it. We're using them as a perfect case study, you see, to understand the fundamental rules of how some diseases can spread so silently and so effectively through a population.
So let's begin with a very important question, something that is critical for public health. Just think about it for a moment. Why are some infections so incredibly difficult to contain? What makes one enemy so much harder to fight than another? Well, the answer, as we will see, is all in their strategy.
Now this is really interesting. Have a look. This is like their playbook, their common strategy. See, they rely on direct person-to-person contact. Why? Because they don't survive well in the environment. They're actually quite fragile outside the body. And this is the most important point. They are often transmitted by asymptomatic people who don't even know they are sick. This asymptomatic transmission, this is the absolute key to their success, and a massive, massive challenge for us in medicine.
All right, let's move on. Next up is a story we all know far too well, COVID-19. But let's look at it from our perspective, okay? Through the eyes of a pathologist, to really understand the mechanics of an acute viral infection.
Just look at this timeline. It shows just how incredibly fast this new virus took over the entire world. Think about it. From a small cluster of pneumonia cases in one city to a global pandemic declared by the WHO in just 3 months. The speed was just unbelievable.
And this number, 6.9 million. This shows the absolutely devastating impact it had globally. And please remember, this is the reported number. It's very likely a big undercount. This is exactly why we need to understand the pathology of this virus and others like it, so we are better prepared next time.
So what happens once the virus gets inside? This is the crucial point. It's not just about the virus you see. It's about our body's response. The infection can go down 2 main paths. Path number 1, our immune system is strong. It controls the virus early, the viral load stays low, and the person gets asymptomatic or maybe just a mild disease. But path number 2, if that control is not good, suboptimal, the virus just replicates like anything, and our immune system can go crazy. It overreacts, causing that devastating cytokine storm we have all heard so much about. But how does the virus manage this?
How does it achieve such a high viral load in some people? How does it win that first critical battle? Well, let's look at how SARS-CoV-2 is so good at evading our immune system. It's truly a master of disguise. Think of the virus like a spy, okay? A spy with perfect fake documents. First, it puts a little cap on its RNA, a disguise, to make it look like our own cell's RNA. So our cell thinks, oh, this is one of us. Then it hides inside a little bubble, a vesicle, while it's replicating, so it's hidden from the cell's alarm systems. And finally, if that wasn't enough, its proteins actively go and block the alarm signals themselves. They stop the production of interferons, which are our body's emergency signals. It's a 3-level deception plan. Very, very clever. Okay, now let's change gears a bit. Let's look at some old enemies, the childhood classics, measles and mumps. These viruses have been with us for a very long time. They are also acute infections like COVID, but their strategies are a little different, and we can learn a lot from them. Everyone knows this, right? The classic sign of measles, this blotchy reddish-brown rash. It's very characteristic, starts on the face and then spreads down the body. But for us as doctors and pathologists, this is what we look for. Under the microscope, these are Warthin Finkeldiei giant cells. Just look at them. They are huge, multinucleated cells, and seeing them is definitive proof. This is what we call a pathognomonic sign. Remember that term, pathognomonic. It means if you see this, it is measles. It's the true signature.
And this is a very important clinical point for all of you. The real danger of measles is often not the virus itself. The big problem is that it causes profound immunosuppression. It basically knocks out the patient's immune system for a while, leaving them completely vulnerable to other infections, secondary bacterial and viral infections. That's what can be so deadly.
Now what about mumps? It's a paramyxovirus, same family as measles, but it has a totally different strategy. No widespread rash here. It specifically targets the salivary glands, right? Causing that classic parotitis, the swelling. And for post-pubertal males, it can have a very serious complication, orchitis, which is inflammation of the testes. Very different target. Okay, now for what I think is one of the most brilliant and cunning strategies in the entire viral world, hiding and waiting. We call these viruses the sleeper agents. So what is a sleeper agent virus? What is this latency? Basically, the virus infects you, but then instead of getting cleared by your immune system, its genome, its genetic material, just goes into hiding. It hides inside your own cells, often in neurons where it's safe, and just stays there. Dormant, silent, sometimes for your entire life. And we see the result of this all the time. The common cold sore or fever blister. This is a perfect example of reactivation. This is not a new infection, okay? This is the old herpes simplex virus that was hiding, sleeping in the nerve ganglia, waking up, and then traveling back down that same nerve to cause a sore on the skin. But again, where do we come in? Under the microscope, we see its footprint, its signature. These are called Cowdrey type A intranuclear inclusions. Can you see how the viral material forms this large, glassy-looking blob inside the nucleus? And it pushes the cell's own DNA, the chromatin, right to the edges. This is a classic telltale sign of a herpes virus infection. And this is the most brilliant illustration of latency. The varicella-zoster virus, VZV. Think about this. The same exact virus causes 2 completely different diseases. First, as a primary infection in childhood, it causes chickenpox, that widespread itchy rash. Then the virus goes to sleep. It goes latent in the dorsal root ganglia for decades. And then, when it reactivates, maybe when the person is older, it causes shingles, a very painful localized rash that follows one single nerve path, or dermatome. It's amazing. 1 virus, 2 diseases, separated by a lifetime. So, okay. We've gone through all these different viruses. What are the key takeaways for us? What are the big lessons we should learn from these microscopic battles? The first thing is that viruses are not all the same. They are not just simple mindless things. They have evolved very different master plans for their survival. Some, like COVID or measles, they use an acute transient strategy. Get in, replicate very fast, spread to the next person, and then get cleared. Hit and run. But others, like the herpes viruses, they play the long game. They use latency, hiding inside our cells for years and years to make sure they survive. You see, understanding these patterns, their attack strategies, their evasion tactics, their transmission methods, this is the absolute key to both medicine and public health. We have learned so much from measles, from COVID, from herpes. But this last question, this is the one that keeps people like me, virologists, pathologists, awake at night. What will the next virus teach us? What new tricks will it have in its bag? And more importantly, will we be ready for it? Something to think about. Okay, thank you. Good morning, future doctors. Settle down, settle down. Welcome. Cello, let's begin today's session. You see, every single day, you and I, we are going into battle. But our enemy, it's not something you can see. It is microscopic, and let me tell you, it is absolutely brilliant. Before we can defeat our enemy, we must first learn to respect it. So this is the fundamental question, isn't it? I want you to really think about this. How does a single-celled organism, a tiny little virus, manage to bring down a system as complex, as magnificent as the human body? Well, it's not luck. It's not by chance. No, it is pure strategy. It's an intelligence that has been sharpened over millions of years of evolution. And that is our job today, class. We are going to open their playbook, we're going to study their tactics, expose their secrets. I want you to think of yourselves not just as doctors, but as detectives, as intelligence officers. We are here to profile the enemy, to understand their moves, so we can plan our counterattack. Okay, let's dive right in. Our first case file. This one is about the spies, the assassins. These are the pathogens that don't meet you on an open battlefield. Oh, no. They are far, far more clever than that. These are the intracellular hiders. You see, their entire strategy, their whole game is built on one simple principle: hide in plain sight. They are hijackers. They take over our own cellular machinery, turning our cells into their own personal factories, their own hiding places, completely safe from the patrolling soldiers of our immune system. It's a truly cunning approach, you have to admit. And here we have one of them, caught right in the act. Our culprit is cytomegalovirus, or CMV. You see that large, dark spot inside the nucleus with that clear ring around it? That is the classic pathognomic owl's eye inclusion. When you see this under a microscope, and you will, you know CMV has been there. It's like the villain's calling card left right at the crime scene for you to find. But listen, this hiding, it is not a passive act. This is active sabotage. I mean, just look at these tactics. First, CMV shuts down the MHC molecules. Think of MHCs as the windows of the cell. They show the outside world what's happening inside. So CMV boards up these windows, making the infected cell invisible to our T-cell police force. Then, it produces fake immune signals, basically creating false radio chatter to confuse our entire response system. It even blocks our natural killer cells, our special forces, and puts on a special coat to hide from antibodies. This is not just hiding. This is sophisticated, high-level espionage happening inside our bodies. Now, for our next case file. You see, not all pathogens are spies. Some are heavy artillery. They don't need to hide when they can simply blast their way through all our defenses. So let's meet the toxin titans. Their strategy is pure, unadulterated brute force. Forget hiding inside one cell. Their goal is to cause widespread chaos and destruction, and they do it using incredibly potent biochemical weapons. These are the terrorists of the microbial world. Just let that sink in for a moment. A single molecule. It's almost unbelievable, isn't it? One single molecule is all it takes to completely shut down a cell's protein factory, and that leads to cell death. That is efficiency in its most terrifying form. And the anthrax toxin is a perfect example of a masterfully designed weapon. It's a three-part system, almost like a guided missile. First, you have the protective antigen. That's the targeting system. It binds to the host cell. Then it creates a channel, basically blowing a hole right in the door. And through that door, it unleashes its warheads, edema factor or lethal factor. A beautiful, deadly, and elegant mechanism. And if you think that's bad, some toxins are even more insidious. Take the super antigens from Staphylococcus. They don't just kill cells one by one. No, they trick our own immune system into launching a massive, uncontrolled, self-destructive attack. It's like turning our own army against our own cities. This leads to a cytokine storm and toxic shock. Truly diabolical. And this is the result. This is not a clean surgical strike. This is scorched earth. A lung abscess caused by staph. You can see the complete devastation, the liquefaction of the tissue itself. This is the calling card of a toxin titan, and this is exactly what you will be seeing in your patients. Remember this. On to our final case file for today. We've seen the spies, and we've seen the bombers. Now, let's meet the magicians, the shapeshifters, the masters of disguise. So let's think about this very simply, okay? Our immune system, it's basically like a police force. It learns to recognize the face of an intruder. We call these antigens. Once it knows the face, the next time that intruder shows up, our police force can mount a quick and effective response. Simple enough, right? But here is the brilliant move. What happens if the criminal can change his face? Every day, a new disguise. Well, the police are always one step behind, aren't they? They're busy looking for someone who doesn't even exist anymore. This incredible strategy is called antigenic variation. And Neisseria is an absolute genius at this. It has a whole library of genes for its pili. Those are the little grappling hooks it uses. And it's constantly shuffling them like a deck of cards, creating a new face every time. For other proteins on its surface, it just adds or deletes bits of DNA. This causes a frame shift that results in a totally new protein. It's a genetic master of disguise, always staying one step ahead. So, we have seen the playbook of the pathogen, the hiders, the bombers, the shapeshifters. Now, what about our playbook? What exactly is the doctor's playbook? And it really comes down to two completely different philosophies of attack, doesn't it? There's the quiet, insidious, stealthy work of an intracellular hider like CMV, and then there's the loud, violent, all-out assault of a toxin producer like staph. Two different strategies, both incredibly effective. And to treat them, you need two completely different counter-strategies. So remember this, the first rule of our playbook: never, ever underestimate your opponent. Whether they use stealth, brute force, or disguise, you must understand that they come prepared. They always have a plan to survive and thrive at our expense. And this is why we are here today. This is why this lecture is so very important. Because by understanding their playbook, piece by piece, we can write our own. The playbook of healing. For the hiders, we choose antibiotics that can get inside ourselves. For the titans, we use antitoxins to neutralize their weapons. For the shapeshifters, we develop vaccines that target the parts of them that can't change. Knowledge, my dear students, is our first and most powerful medicine. So I will leave you with this one thought to take with you. Now that you have seen their tactics, their genius, which pathogen do you find the most brilliant in its deadly elegance? The spy, the bomber, or the shapeshifter? Think about it, because understanding their genius is the very first step to outsmarting them. Class dismissed.
Namaste everyone, welcome back. Today we're going to do something a little different. Forget the usual lecture. Think of this more like a detective's briefing. We're going to profile some of the most notorious criminals you will ever study. So cello, let's get started.
So I want you to put your textbooks away for a second. Today, you and I, we are criminal profilers. Our job? To unmask these microscopic gundas, these villains of medicine. We're going to look at everything, their hideouts, their weapons, and most importantly, their modus operandi, their method of attack. Why? Because if you understand how the enemy fights, only then can you figure out how to defeat them.
All right, first up on our most wanted list are what I call the aggressive invaders. These are the thugs of the microbial world. No subtlety, no hiding. Their game is all about brute force. They hit fast, they hit hard, and they create absolute chaos. A real shock and awe attack on the body.
And who is our prime suspect in this category? Well, it's none other than the infamous Yersinia pestis. Yes, the agent of plague. This little bacterium is a legend in the worst possible way. Think about it. The Black Death in the 1300s. That was this guy. Its reputation is absolutely earned. This is a true stone-cold killer.
So how does it do so much damage? What's its main funda? It has this incredible weapon. It's called a type 3 secretion system. Now, the easiest way to picture this is like a tiny microscopic syringe. The bacterium literally sticks this syringe into our own immune cells and injects a cocktail of toxins.
These are called YUP proteins. And what do these YUPs do? They are saboteurs. They completely paralyze the cell. They block its ability to, eat the bacteria, what we call phagocytosis, and they cut the communication lines so the cell can't call for backup. It's a brilliant strategy, really, but absolutely deadly.
So you see what happens next, right? With our body's first responders completely neutralized, Yersinia just multiplies like crazy. It goes completely unchecked, and the result is pure chaos inside the body. We're talking massive bacterial growth, tissue necrosis, widespread hemorrhage, and of course those classic painful swollen lymph nodes that we call buboes. That's where the name bubonic plague comes from.
Okay, let's move on to villain number 2 in this category. This one is a bit different. It's an opportunist, a real menace, especially in hospitals. I'm talking about Pseudomonas aeruginosa. Now, this one isn't causing massive pandemics, no. Instead, it preys on the vulnerable. It targets patients who are already weak, especially those with severe burns or patients with cystic fibrosis, whose lungs are already in a compromised state.
Now, let's look at the evidence. This is the crime scene. What you're seeing here is Pseudomonas' calling card. It has a special preference for attacking the walls of our blood vessels. You see that purple-blue haze? That's a huge mass of bacteria. They're causing vasculitis, inflammation of the vessel, and leading to thrombosis, which is clotting. So it's a direct attack on our body's supply lines, literally choking them from the inside.
All right, let's shift gears. We've seen the thugs. Now let's meet a different kind of criminal entirely. This one isn't a brute. This is a master of disguise, a long con artist. I'm talking about syphilis, caused by Treponema pallidum. There's a reason we call it the Great Mimicker. It can hide for years, even decades, and then reappear in so many different forms that it can fool even the best doctors.
And here it is, the culprit itself. Just look at that shape, that slender, corkscrew design. That's not just for show, it's a key part of its strategy. It allows this spirochete to drill and move through our tissues with incredible ease. Plus, it has an outer sheath that basically hides its antigens. It's like wearing a cloak of invisibility, making it so much harder for our immune system to spot it and grab onto it. Very, very sneaky. The whole con, this deception of syphilis, it plays out in 3 acts, like a play. Act 1, the primary stage. A single, painless ulcer, the chancre, shows up and then it just disappears. Poof, gone. Then, weeks later, Act 2 begins, the secondary stage. You get rashes, classically on the palms and soles, and these infectious plaques we call condylomata lata. And then, that also disappears. But, and this is the crucial part, the bacteria are not gone. They've just gone into hiding. They enter a latent period that can last for years, even decades, before the final devastating act, tertiary syphilis, where it comes back to attack the heart, the brain, and other organs.
So you have to ask, what is the underlying mechanism here? What connects all these different stages? It's one key pathological process, proliferative endarteritis. Okay, what does that mean? Basically, the spirochete causes this long-term inflammation inside our small arteries. The walls of these arteries get thicker and thicker, and the opening gets narrower and narrower until the blood supply is choked off. That's ischemia. And that ischemia is what's causing all the damage we see, right from that first chancre all the way to the fatal damage to the aorta in the tertiary stage.
Okay, on to our final category of villains. And these are the patient ones, the silent infiltrators. They don't need brute force, they don't need fancy disguises. Their entire strategy is based on one thing, getting inside our own cells, our own immune cells, the macrophages, and then just hiding, building their empire quietly from the inside. And of course, the masters of this strategy are the mycobacteria.
And the king of this category is without a doubt tuberculosis. Now, it's absolutely crucial for you to understand the 2 main forms. First, there's primary TB. This is what happens when you're exposed for the very first time. Now, in most healthy people, the immune system is strong enough to wall it off and contain it. It just leaves behind a little scar, what we call a Gong complex. But the bacteria can lie dormant, and if your immunity weakens, maybe years later, you can get secondary TB. This is a reactivation of that old infection. And this time, it's much, much more destructive. It loves to attack the apex of the lungs, and this is where we see those big cavities form. And here, this is what that destruction looks like. This is along with advanced secondary TB. Look closely at the upper lobes. See those huge ragged holes? Those are the cavities. And that crumbly yellowish white material? It looks like cheese, right? That is caseous necrosis. This is the pathological signature of TB. The lung tissue is literally being destroyed, eaten away.
So how does our body even try to fight this?
It builds a jail. That's what you see here, the granuloma. The best way to think about it is a microscopic prison. Our own immune cells, the macrophages and lymphocytes, they come together and form a tight wall around the TB bacteria to try and contain them. This little structure is the central battlefield. The entire war between the bacteria and our body is happening right here, inside this granuloma.
Now this slide shows you everything. It's the whole story in one picture. On the left, you see a strong immune response. The granuloma, the jail, is working. You can see a few of the red-stained bacteria, but they are contained. Now look at the right. This is what happens in an immunocompromised patient. The jail walls have crumbled, the bacteria have broken out, and they are multiplying like mad inside the very cells that are supposed to kill them. The entire outcome of a TB infection, it comes down to this one battle.
And this idea, this theme of host immunity being the deciding factor, it becomes even more important, even more central when we talk about TB's cousin, Mycobacterium leprae, the bug that causes leprosy. Because with leprosy, it's fascinating. The disease you get is determined almost entirely not by the bacterium, but by your own immune response.
So, think of leprosy as a spectrum with two opposite ends, two poles. At one end, you have tuberculoid leprosy. This is when the patient has a really strong T-cell response, a Th1 response. The body builds good granulomas to contain the infection, so you find very few bacteria. We call this paucibacillary, pauci meaning few. Now at the complete opposite end, you have lepromatous leprosy. Here the Th1 response is very weak. The body fails to contain the bacteria and they just multiply into huge numbers. This is multibacillary, multi meaning many.
And you can literally see this difference under the microscope. It's amazing. Look at the left. That's tuberculoid leprosy. You see those well-formed granulomas? They're doing their job. But notice, they're attacking a nerve bundle. So a strong immune response, yes, but it comes with a cost, collateral damage. Now look at the right. That is lepromatous leprosy. Just look at those macrophages. They are absolutely stuffed to the brim with red-staining bacteria. It's a total failure of the immune system to contain the infection.
Okay. So we've profiled these different felons, each with their own unique style. What have we learned? What are the big lessons from this battlefield of infectious disease that you need to take with you?
Well, first, we've seen a whole range of attack strategies, right? The modus operandi of each is different. You've got Yersinia with its direct injection of toxins. You've got Pseudomonas going straight for the blood vessels. Syphilis, the master of disguise and immune evasion. And of course, the Mycobacteria with their stealthy strategy of hiding inside ourselves, forcing our body to build those granuloma prisons. Every villain has a unique playbook.
But if there is one central theme, one big idea that connects all of these, what is it? It's this. Especially with those insidious infiltrators, the patient's own immune response, and specifically their T-cell-mediated immunity. It's often the single most important factor. It's what decides whether you contain the infection or whether the infection wins.
And that leaves us with one final thought to ponder. You see, this is a constant arms race. These microbes, they aren't just sitting still. They evolve. They develop resistance to our best antibiotics. So my question to you, the next generation of doctors, is this. Now that you understand their strategies, now that you know how crucial the immune system is, how do we evolve our strategies? How do we get better at diagnosing, at treating, and at preventing these diseases to stay one step ahead? That's what I want you to think about. All right, that's all for today. See you next time.
Hello, students. Welcome back. Today, we're going inside the body to look at the secret strategies of some of our smallest but most dangerous enemies. So, ever wondered how a teeny tiny bacterium or a fungus can cause so much trouble in the body? It's not just random bad luck, you know? It's all about their game plan, their playbook for infection. Okay, let's dive into this. We are going to explore 3 brilliant but absolutely deadly strategies that these pathogens use to outsmart our body's defenses. We've got the masters of disguise, the agents of chaos, and the silent strikers. We are framing this whole thing as a playbook, right? Where each type of pathogen has a unique set of moves to establish an infection and cause disease. First up, we have the real masters of deception, the spirochetes. These guys are smart, y'all. They play a long, multi-stage game that can fool the body for years, even decades. Let's start with a classic example, syphilis, caused by Treponema pallidum. This infection literally changes its face over decades. It's the ultimate disguise artist. And this step-by-step progression is the key to its strategy. See, it attacks with a small, painless chancre that you might even ignore. then it seems to retreat. But then it comes back with a widespread rash as it spreads. And finally, years later, it returns with devastating force, causing damage to the aorta, the brain, everything. Now, what you're seeing here is the long-term consequence. This is heparoblast. This severe scarring on the liver is caused by what we call gummas, which form in tertiary syphilis. It really shows you just how destructive that final stage can be. And syphilis is not alone in this game, okay? Lyme disease, caused by another spirochete, Borrelia burgdorferi, follows a very similar multi-stage playbook. The big difference? This one is delivered by a tick. And this timeline just perfectly illustrates the whole story. It starts with that classic bullseye rash, erythema migrans. But from there, weeks to months later, the bacteria spread through the blood, causing pain, meningitis, even heart problems. And then much later, months to years down the line, it leads to chronic arthritis and can even cause a debilitating encephalopathy, all from one tick bite. Okay, let's move on. Our next strategy is completely different. This is not about hiding. No, this is about deploying incredibly potent toxins for a rapid, chaotic assault. This is pure chemical warfare, and the genus Clostridium are the experts. Now, this is fantastic. You have to appreciate this. 2 toxins from the same family of bacteria causing the absolute opposite effects. Look here. Tetanus toxin blocks the stop signal, the inhibitory neurotransmitters like GABA. The result? Spastic paralysis. Your muscles lock up. Then you have botulinum toxin. It blocks the go signal, acetylcholine. The result? Flaccid paralysis. Your muscles can't contract at all. One won't let you relax, the other won't let you contract. Brilliant, isn't it? And here is another member of the Clostridium family, Clostridium perfringens. Look at these boxcar-shaped rods. This one uses a different kind of chemical warfare to cause something truly terrible called gas gangrene. So the crucial point here is that this bacterium doesn't just make you sick, it physically dissolves your tissues with powerful enzymes. It causes myonecrosis, which is just a fancy term for muscle death, and it produces gas within the tissue, leading to this rapid, widespread destruction, a true agent of chaos. All right, our final strategy. This one is the ambush. These pathogens are often harmless, yeah? They live on us, around us, just lying in wait. They are the silent strikers, waiting for the perfect moment to attack, right when our defenses are down. And this quote, it gets to the heart of the strategy. Candida albicans is a commensal organism, a normal part of our microbiota. What does that mean? It means the enemy is already inside the gates, just waiting for an opportunity. Here we can see the disease at 2 different scales. On the left, look at that widespread infection in the esophagus, those horrible white plaques. That's the gross pathology. And on the right, under the microscope, that's the culprit. You can see the characteristic pseudohyphae and the budding yeast forms of Candida. And this is exactly why we call them opportunistic. They are waiting for an opportunity. They exploit a weakness in the host's immune system to launch their attack. In a healthy person, no problem. But in patients with neutropenia, AIDS, or on chemotherapy, that's when they strike. And look at this incredible image. This is another silent striker, Cryptococcus. It can invade the brain of an immunocompromised patient and create these classic soap bubble lesions. Can you see them? The gelatinous masses of the fungi are literally expanding the perivascular spaces. It's a truly scary sight. So, to wrap this all up, let's just quickly review the 3 distinct plays we've seen from this pathogen playbook. And remember, this isn't just for your exams. Understanding this is about being a good doctor. This table here is a perfect summary. Strategy 1, the masters of disguise, like Treponema pallidum. Strategy 2, the agents of chaos, like Clostridium, causing rapid damage with their toxins. And strategy 3, the silent strikers, like Candida, attacking only when our defenses are weak. So you have the long con, the chemical assault, and the ambush. So 3, just remember these 3 fundamental strategies. If you can understand these patterns, it is the first step to diagnosis and treatment. You know, when you think about it, from the slow deception to the rapid toxic shock and the sneaky ambush, each strategy is really a marvel of microscopic evolution. It's an arms race between us and them. And why does this matter so much? Because understanding the how helps us predict the what. If you know the enemy's playbook, you can anticipate their next move and develop better ways to fight back. This is what being a doctor is all about. This leaves us with a final thought, doesn't it? This battle is never over. As medicine advances, what new plays, what new tricks will these pathogens add to their playbook next? That's the question for your generation to answer. Thank you.
All right, everyone, settle down, settle down. Welcome back. Today, we're diving deep into the world of our most cunning, microscopic enemies. Now, this isn't just theory for your exams, okay? These are the invisible invaders you will be fighting for your patients your entire careers. So pay attention, because this is a battle you absolutely need to know how to win.
Let's start with a bang. Look at this. This is not some abstract painting. This is war, happening at a microscopic level. What you are looking at is a blood vessel inside a human brain. And those ribbon-like things? That is a fungus, mucor, literally tearing its way through the vessel wall. This is the reality of the fight we're up against. The stakes don't get any higher than this.
Now, the fascinating thing about mucor is that it is a true opportunist. What does that mean? It means it's everywhere, in the dust, in the air, just waiting. In a healthy person like you or me, it does nothing. It's like a thief that only attacks when you leave the door wide open. It waits for the body's defenses to go down, and only then it strikes.
So how does a tiny invisible spore become a deadly invasion? It's a four-step story. First, we just breathe in the spores. Simple. Our macrophages, the first-line soldiers, try to gobble them up. But if some escape and start to grow, our neutrophils, the second line of defense, come in to attack. But if both of these defenses fail, the fungus wins. It breaks through, gets into the blood vessels, and causes that horrible tissue death we just saw. Okay, now this is a Viva favorite, so listen carefully. For your exams, and more importantly for your patients, you must know the difference between mucor and its cousin, aspergillus. It's all about the branching angle. Look, mucor has these broad hyphae with no cross walls, and they branch at a perfect 90-degree angle. Think of a right angle, like the corner of a square. Aspergillus, on the other hand, has cross walls and branches at a sharp, acute angle, more like a V. This small detail, this is the clue that tells you what you're fighting.
And this is where it all clicks together. Why was black fungus such a problem in diabetic patients during the COVID wave? Simple. This fungus loves iron. It needs it to grow. And in patients with diabetic ketoacidosis, or DKA, their body's metabolism is in chaos, and it releases a lot of free iron into the blood. It's like we are setting up an all-you-can-eat iron buffet for the fungus. And believe me, it eats very well.
All right, next up, we've seen a brute force attacker. Now let's meet a different kind of fungus, one whose strategy isn't strength but deception. Meet histoplasma, a true master of disguise.
We call histoplasma the great imitator, and you need to know why. Because it presents with fever, cough, night sweats, and coin lesions on a chest x-ray. Now, what does that sound like? Tuberculosis, right? In a country like India where TB is so common, your first thought will always be TB. But a good doctor thinks beyond the obvious. You must remember this mimic. It might just be a fungus fooling you.
This is brilliant. It shows the two faces of the same disease, and the only difference is the patient's immune system. On the left, a healthy person. Their immune cells surround the fungus and lock it up in a little calcified gel called a granuloma. No problem. But on the right, an immunocompromised patient, someone with HIV maybe. Without a strong immune system, the fungus just takes over the cells and spreads everywhere. Same bug, two completely different stories. Okay, we're done with fungi for now. Let's switch gears completely and move into the world of parasites, the protozoa. And of course, we have to start with the most infamous, the biggest killer of them all, Plasmodium, the parasite that causes malaria.
Before we get into the biology, I want you to just look at this number, 619,000. That's almost 6.2 lakh people dead in just 1 year. These are not statistics for you to memorize. These are lives, many of them children. This is why you are sitting here. This is why we have to understand this disease inside and out.
This life cycle is the key to everything. You must know it by heart. It starts with a mosquito bite. These little sporozoites get injected, go for a holiday in the liver, and multiply quietly. The patient feels nothing. Then they burst out and invade the red blood cells. Inside the RBCs, they multiply again. And here's the crucial part. When all these millions of infected cells rupture together, that's what causes the classic chills and high fever. The patient's symptoms are literally a clock, ticking to the parasite's schedule.
Now, not all malaria is the same. There's one species that is the true gangster, Plasmodium falciparum, Why is it so much deadlier? 3 reasons. 1, it's not picky. It infects red blood cells of any age, so the number of parasites gets very, very high. 2, it makes the infected cells sticky, so they block up small blood vessels. And 3, it triggers a massive immune overreaction, a cytokine storm, that causes huge damage. These 3 things make falciparum malaria a medical emergency.
And this is the ultimate result of that stickiness. What you are seeing is a tiny blood vessel in the brain, completely choked, completely blocked by infected red cells. No blood can get through. No oxygen can get to the brain. This is cerebral malaria. This is what causes confusion, coma, and death. It's the most feared complication, and it's absolutely devastating.
You know, as doctors, we have to respect our enemies, and the survival strategies of these parasites are just incredible. They have evolved for millions of years to outsmart us. Plasmodium is a master, but it's not the only one. Let's look at a few other parasites and their unbelievable tricks.
Just look at this slide. That big cell is a macrophage. It's the commando of our immune system. Its only job is to hunt and kill invaders. But look inside. It's packed full of those little dots. That's Leishmania. This parasite has done the unthinkable. It has turned the police station into its own safe house. It's living and multiplying inside the very cell that was sent to destroy it. It's just amazing.
And it's not just hiding, it's actively redecorating the place to make it more comfortable. It cuts the phone lines so the macrophage can't call for help, it disarms the internal bombs, it even changes the acidity inside the cell's stomach to create a nice, cozy home for itself. It's an absolute master of subversion.
So, Leishmania is a hider, a master of stealth. But this next parasite, the trypanosome that causes sleeping sickness, it does the exact opposite. Look at it, swimming freely in the bloodstream. It's not hiding at all. It's right out in the open, exposed to all our antibodies and immune cells.
So this leads to the obvious question, doesn't it? How on earth is it surviving out there? The blood is a war zone, filled with antibodies that should be destroying it. What is its secret? Well, its secret is a work of pure genetic genius. The answer is something called antigenic variation. It's like a master of disguise. Imagine the police are looking for a thief in a red coat. Our immune system makes millions of red coat antibodies. But just as we're about to catch all the thieves, a few of them quickly change into a blue coat. Now all our red coat antibodies are useless. The blue coat thieves multiply until we make new antibodies, and then they change coats again. Each wave of fever the patient gets is the parasite changing its coat. Brilliant, isn't it?
So for all their clever tricks, here is the good news for us. Every one of these pathogens leaves behind a fingerprint, a visual clue, a calling card that we as clinicians can find under the microscope. Every slide you look at tells a story, if you just know how to read it.
Here's a perfect example. Look inside that red blood cell. Do you see it? That perfect arrangement of 4 parasites in a cross shape. This is the famous Maltese cross of Babesia. This is what we call pathognomonic. A fantastic word. It means if you see this sign, the diagnosis is 100% confirmed. Game over. You found your culprit. And here is another one of those unique fingerprints. This large, thick-walled sphere, just packed with tiny little dots inside. This is a sphereal of the fungus coccidioides. It looks like nothing else. You see this in a biopsy from the lung, you know exactly what you were dealing with. These are the visual clues that make you a great diagnostician.
And I will end on a very, very important clinical warning. Look at this. It's an intestinal biopsy, and it is absolutely swarming with worms. This is strongyloids hyperinfection. And you know why it happened? Because the patient was given high-dose corticosteroids, a common drug. But by suppressing their immune system, we woke up a sleeping monster that was living quietly inside them. The lesson is, always be careful. Your treatment can sometimes unleash a hidden enemy.
So, we have seen their strategies, their disguises, and the terrible damage they can do. The patient in front of you is the battlefield. This knowledge that you're learning here, this is your weapon. The only question that remains is how will you use it to win the next battle? That's the challenge you are going to face every single day. Study well.
Hello, Butchels, and welcome to class. Today, we're going to play detective a little bit and investigate some of the smartest criminals in the microscopic world. I'm talking about parasitic worms, or helminths. You know, these aren't just some simple parasites living in the body. No. They are master manipulators that can actually turn our own body against us. And that brings us to the big question for today, our central mystery. Just look at this picture. How on earth is it possible that a creature, sometimes so small you can't even see it, can cause such a massive, devastating change to a human leg? This is the question we are going to solve together today. So I want you all to be thinking about it. All right, so here's our plan of attack for today. First, a little briefing on these unseen invaders. Then, we're going to open 3 very important case files. One on a worm that lives in our muscles, another whose eggs are the real problem, and a third that's a master of blocking our internal drainage system. And finally, we'll see what we've learned from this battlefield, which is our own body. Let's get started.
Okay, now this is absolutely the most crucial point to understand right from the beginning. With many of these worm infections, the worm itself is, well, it's not the main problem. The real damage, the most severe disease, actually comes from how our own immune system reacts to it. It's the collateral damage from the war going on inside us that causes all the major pathology. Right then, let's open our first case file. Our culprit here is Trichinella spirillis. Now, this is a very sneaky parasite. It gets to us through something we all do every single day, just eating a simple meal. So how does this fellow get inside? The main entry route is by eating its larvae in undercooked meat. In India, and really many parts of the world, pork is a very common source. But you can also get it from game animals like wild boar or even bear. Basically, any animal that eats meat can be part of this chain. The life cycle is actually quite straightforward. We eat the larvae in the meat. Inside our gut, they grow up into adults, they mate, and then the female worm releases thousands of new larvae. These tiny little larvae then dig through our tissues, get into our bloodstream, and travel all over the body. But where are they going? Well, their final destination is our skeletal muscle cells. That's where they set up a permanent home. For this particular worm, humans are basically a dead end. We're the end of the... And here it is, the culprit, caught red-handed. Botchel. This is what it looks like under the microscope. This is a slide of skeletal muscle. You can see these pink muscle fibers, and right there, inside them, you see those coiled up larvae? Each one is inside its own little cyst. It has literally burrowed into our own cells to live there. A true intracellular parasite. So, when a patient with this comes to you, what will they look like? Remember this classic presentation, it's very important. The patient will have a high fever, they'll have severe muscle pain, which makes sense, right? The worms are living in the muscles, and this is very, very characteristic, swelling around the eyes. We call it periorbital edema. And when you look at their blood report, you will see a sky-high eosinophil count. That's a huge clue for any parasitic infection. In very bad cases, if the larvae decide to go to the heart or the brain, it can even cause life-threatening heart failure or encephalitis. Okay, let's move on to our second case file, schistomyosis. This one is found in tropical freshwater and it's spread by snails. The adult worm actually lives in our blood vessels, but it has a secret weapon, something that causes way more destruction than the worm itself. This sentence right here, this is the absolute key to understanding schistosomiasis. The adult worms, they're the trigger, but it's the thousands upon thousands of microscopic eggs they release that are the real weapons. These eggs, you see, they get trapped in tissues like the liver, and, well, that's where all the trouble really begins. This slide, it tells the whole story perfectly. On the left, you see the cause. Those pink oval things are the schistosome eggs, trapped in the liver. And all that stuff around them, that's a huge collection of inflammatory cells called a granuloma. It's our body trying to wall off the egg. Now look to the right. That's the long-term effect. Over years and years, thousands of these healing granulomas leave behind massive scars, what we call fibrosis. And this scarring literally chokes the liver's blood supply. This specific kind of scarring has a very unique name, pipe stem fibrosis. Please remember this name, it's a very high yield point for all your exams. The fibrosis, it forms around the portal veins in the liver, making them look just like the stem of an old clay pipe if you were to cut the liver open. This blocks the blood flow, of course, causing a massive backup of pressure we call portal hypertension, which leads to all sorts of dangerous complications. Now, it's also critical to know that not all schistosoma species are the same. In parasitology, location is everything. So S. mansoni and japonicum, they like to live in the veins around the liver and the gut. So naturally, they cause that pipe stem fibrosis we just talked about. But S. hematobium, it prefers the veins around the urinary bladder. So its eggs get trapped in the bladder wall, causing bleeding, obstruction, and, this is very important, a much higher risk of squamous cell carcinoma of the bladder. Okay, time to open our final case file for today.
The filarial worms. These are microscopic, thread-like worms that are basically architects of internal blockades. And who is their accomplice in this crime? The very common, very humble mosquito. And here we are. This is the answer to that question I asked you at the very beginning. This is elephantiasis, a truly shocking and debilitating condition, and it's the end stage of a chronic infection with filarial worms like Wucheria bancrofti. This is exactly what happens when the body's entire drainage system gets completely blocked up.
So, how does it get this bad? Well, it's a very slow, step-by-step process. The adult worms, they move in and take up residence inside our lymphatic channels. Just their presence there causes chronic inflammation, which damages the walls of these vessels. Over many, many years, this leads to scarring, fibrosis, and a complete blockage. Now, the lymph fluid, which is supposed to be draining from the tissues, has nowhere to go, so it backs up, causing this massive swelling, or lymphedema. And over time, the skin on top of it becomes thick, hard, and pebbly.
But wait, the story of filarial worms doesn't just end with blocked lymphatics. Another species in this family, Onchocerca volvulus, has a completely different target. It attacks the eyes and causes a condition that's tragically known as river blindness. You see, the adult worms of Onchocercosea live all knotted up together inside these fibrous nodules under the skin. We call them onchocercomas. You can actually feel these lumps on a patient. But just like we saw with schistosomiasis, the adult worm isn't the main villain here. The real damage is done by what it produces. So, this is the crucial distinction you have to make. The adult worms, they just sit quietly in their nodules. It's their millions of tiny offspring, the microfilaria, that actually cause the disease. These little larvae migrate restlessly through the skin, causing this maddening, intense itchiness and dermatitis. And when they migrate into the chambers of the eye, they trigger a severe inflammatory reaction that scars the cornea, destroys vision, and leads to irreversible blindness.
So, after going through these 3 different case files, what's the common theme? What have we learned? It becomes pretty clear, doesn't it? In all these infections, our own body is the main battlefield, and it's our immune system's weapons that often end up causing the most damage. Let's just summarize the key take-home messages. First, the most severe pathology is very often collateral damage from our own immune response. Second, processes like forming granulomas and fibrosis, things which are meant to be protective, they're the very things that destroy the function of our organs. Third, these parasites are absolute masters of manipulation. And finally, always remember that location determines the disease. Whether an egg lands in the liver or the bladder, it changes everything. And all of this leaves us with a final, thought-provoking question for you to ponder. In this war against these invaders, is our only goal to kill the worm, or should we also be focusing on finding ways to safely calm down the inflammatory storm that our own body is creating? It's something to think about, isn't it? That's all for today. Thank you, Bacho.